Hi, my name is Yongheng. I'm a postdoc associate working in Professor Manolis' Kellis lab. Currently, I'm building agentic tools to automate single-cell data analysis. So historically, people analyze single-cell data using different Python or R packages. It takes days, months, even years to accomplish or complete a project. Now, using agentic tools, we automate the whole process. Now, users can just give a simple prompt, then we have a lot of things in the backend that will automatically complete the whole process. We're comparing healthy donor versus donors with Alzheimer diseases. By looking at the data, we can know what biomarkers or what gene is associated with the Alzheimer disease. That's one aspect, the biological insight. If we know those biomarkers, we can potentially develop drug to target those biomarkers. For example, if one gene is upregulated, we can potentially design drug to downregulate that gene, potentially mitigate the symptom. So I think the exciting part is virtual cell. People are building kind of simulation that can understand what's happening in the world. For example, you generate a video, they know when you drop something, it should fall to the ground. What we're building is a simulator that understands what happened within a cell, that is to the nanometer level, to the cell level. What's exciting about that is, I will give you one example. Nowadays, when pharmaceutical company try to evaluate the drug effect, they actually need to go in, do the experiment, treat cells with drug. For example, treat cancer cells with cancer drug to see if the drug can actually kill the cells. But I think when we accumulate enough data, we would be able to predict whether a drug will kill the cancer cell without doing any experiment. So the tool I'm building is more for scientists working in biological laboratories. Instead of spending weeks or days analyzing data manually, sitting in front of a computer, they can just click once, tell our software where the data is, and they can just go work on something more innovative and come back a few days later. We provide the figure needed, the discussion of the figure, and in like a manual script format. The complicated part about single-cell sequencing is that we are getting 37,000 gene expression level. I will give you one example to understand that, right? If I stand up, you can see the shadow of myself. That's kind of projecting 3D object to 2D. Imagine you do that kind of projection 1,000 times to kind of reduce the dimension from 37,000 dimension to a 2D plot. The human cognitive limit, we have a problem understanding high dimension data. But computational tools and Gen AI can reduce that dimension for us to understand the data. That is like a human robot or human software interaction that can help us better understand the data, further guide the software to review biological insight for drug development. I think it's very exciting to think about we're building a tool that can help thousands of biologists. Everyone wants to work on something innovative. We don't want to work on things that are repetitive. But currently, we have to spend days, weeks, months on something that we're so familiar with, but the software keeps giving us bugs. We have to solve the bugs by ourselves. Imagine a future where the software can do all that for you. And what you need to do is just provide your knowledge, provide your feedback to the software that will automatically analyze the data, and you can always work on something more innovative. I think that's an exciting future I'm looking forward to.